our speaker's all ready to go with his presentation. So it gives me great pleasure to introduce our speaker today, Professor Ed Bouwer from Johns Hopkins University in Baltimore. And Ed is the Abel Woolman Professor of Environmental Engineering at Johns Hopkins. He did his master's and PhD at Stanford University. And prior to going to Johns Hopkins, he was an assistant professor at the University of Houston. And his research interests are environmental microbiology and waste treatment. So we're very pleased to have him speak to us today. And as you can see on his slide, his co-author is Stephen Chow, who is a graduate student of Ed's, who has participated in some of our IIES events in the past. And with that, I just welcome all of you, whether it's morning, afternoon, or evening, depending where you are. And thank you very much, Ed, for your presentation.
Great.
Over to you.
Thanks, Doug. So greetings from Baltimore. And yes, I'd like to acknowledge Stephen Chow as a co-author. He helped prepare several of these slides, and it's really his research that I'll be talking about later in the talk. So as you see from the title, I'll talk a little bit about groundwater remediation and give some background. Here's an outline of the presentation. And then we'll focus on our contaminant of interest, which are chlorinated benzenes, and talk about a proof of concept that we're working on for a treatment technology that involves both anaerobic and aerobic processes in using a dual biofilm system. And then I'll conclude with some ongoing work for our next steps. So just as background, we've had a series, probably now 30, 40, up to 50 years. We've known that a lot of hazardous waste sites have been around. Superfund, you may have heard of, sort of in the late 70s, early 80s. And so we've been working on a lot of these sites to clean them up, and we've made some progress. I list here basically a recent review showed that 360 sites out of over 1,700 what are called NPL sites or Superfund sites have been so-called cleaned up or closed. A lot of RECRA sites, which are existing operations that had contamination, have controlled human exposure, a majority of those sites. And then there's been an underground storage protection program where over 1.7 million underground storage tanks have been either replaced or closed since 1984. But other sites are very complex, and so there are a lot of sites still remaining. And in this case, groundwater contamination remains difficult, and it can take 100 years or more for these sites to be cleaned up. And these sites are usually characterized by large in size, having heterogeneous hydrogeology, and having a mix of contaminants, some of which are very recalcitrant. So a recent review, 2013, from the National Research Council showed about 126,000 of these sites still remain, and the estimated cost to completion is hundreds of billions of dollars. So here's a little schematic of what one of these complex sites look like. If you release a contaminant, in this case called a dense non-aqueous phase liquid, it'll tend to sink when it gets in water. And if you have a hydrogeology that's complex, you'll get these ganglia and pools of this material. And then as groundwater flows through it, you form a dissolved plume. And then things can even get worse. If there's bedrock at the bottom, which is fractured, this stuff can work its way into the bedrock. And so you really have a notorious issue with all this contamination residing in these pockets, and it's very difficult to remediate that. But nevertheless, a lot of remedial technologies have been developed, and their targets are two primary zones. We either look at the source zone, which is right immediately in the area where the contaminant was spilled, and then we look more down gradient at the dissolved groundwater plume. And so some of the current technologies that deal with the source zones, in situ thermal treatment, basically you heat the ground through either electrical resistance or conductive heating, and this then causes the contaminants either to volatilize or even burn in some cases. You ignite them basically in the soil, and for a hydrocarbon, for example, and you remove the mass that way. Chemical transformation processes using very strong oxidants, zero-valent iron, Fenton's reagent, Fenton's chemistry have been used to oxidize the contaminants. And then extractions using various flushing techniques using co-solvents or surfactants have been employed. So those are where we're looking for source zone. And then when you look at the dissolved groundwater plume, pump and treat has been around for a very long time, where you pump up the water and do some sort of above ground treatment. Unfortunately, it can take a very long time because there's very slow dissolution of the contaminants that way. Another way to at least prevent the risk and the harm is to have physical containment. So basically contain it so no one is exposed to the contaminant. And then we can look at a containment system that has permeable reactive barriers and employ bioremediation, which is really the topic of this lecture, using microorganisms. And then, of course, combine remedies. So you can get very clever and engineer a system to look at both the source zone and dissolved plume treatment. So here's again that schematic. And so some of the technologies are you either can remove the contaminant on the surface and cap it to prevent exposure. Here's pump and treat, where you are pumping out the groundwater and treating it above ground, or at least slowing the movement of that water, putting in some sort of barrier system to prevent the down gradient movement can be done. And then if you have material leaching into the surface water through groundwater intrusion, then that's also an opportunity for contamination and removal. So our work focuses on bioremediation. And so here are just some general principles about bioremediation. It requires appropriate organisms and a favorable biogeochemistry. And fortunately, a lot of these contaminants, even though they're toxic and harmful at low concentrations, many of them can be biodegraded. So we rely on sometimes what's called natural attenuation. This is where if the organisms in the subsurface can naturally degrade the chemical. This often occurs for hydrocarbons, petroleum compounds. They're readily degradable, and so you can basically rely on the native bacteria to do that. But for other contaminants, particularly chlorinated solvents and pesticides, polychlorinated biphenyl compounds, we find that we have to look at what's called enhanced bioremediation, where you actually have to stimulate the organisms that are there. So this requires delivery of electron donors, electron acceptors, or other growth factors like nutrients. So this is called biostimulation. And then in cases where you don't have the right microbe present, you can add microorganisms, and this is called bioaugmentation. And bioremediation has been used as a remedy at about 25 to 30% of Superfund sites. And as I mentioned, applications, petroleum hydrocarbons, chlorinated solvents, pesticides, and using both aerobic and anaerobic processes. And so by doing bioremediation, the idea is that you clean up or you destroy the chemical in the ground, and that way you eliminate exposure pathways. So to reduce risk, you're really looking at reducing exposure pathways. So when we look at exposure to water or volatilization, then this is how humans typically get exposed. So the predominant concern, of course, is human exposure. Now, why I mentioned chlorinated solvents. Why are chlorinated solvents so ubiquitous? Why do we find them so frequently? Well, it turns out that there's a lot of benefits of having chlorinated compounds. These chlorinated solvents have excellent cleaning properties, so that's why we call them solvents to begin with. So they can solubilize hydrophobic chemicals. They're often used as degreasers, paint strippers, stain removers. So they're very good in that way. They also have low miscibility in water. So if you want to use them as an extraction agent, then you can recover the compounds. They have high boiling points, low flammability. So as a solvent, they have very good properties. And then some of them we know, the polychlorinated biphenyls, PCBs, had good insulation properties. So they were used in transformers and electrical systems. And then some chemicals, especially chemicals like deodorants, pesticides, and various intermediates. So we do have widespread use of these compounds, and it's not surprising that in the environment we find that, at least for chemical releases, that these are very commonly found at hazardous waste sites. And so some surveys that have been done, particularly the USGS, has done what they call through their NAQUA program, the National Water Quality Assessment Program. They've identified a lot of regions across the US to try to represent different watershed systems, and they've sampled a lot of wells. And in this little map of the US, the wells that are labeled in red have detected solvents in them. There are a lot of other wells sampled with no detection of solvent. Those are sort of the gray, light blue dots that are shown. But a large fraction of the wells that were assessed were positive. 17% of those reported were positive for chlorinated solvent detection. And so some of the reports that they've done look at $750 billion for groundwater remediation. And then in our case, we're going to start looking at our case study with chlorinated benzenes. The 8% of these Superfund sites have detected chlorinated benzenes, this type of chlorinated solvent. So we're looking at chlorinated benzene compounds. These compounds are sometimes called semi-volatile compounds because some of them are volatile, but they also are dense non-aqueous phase liquid. Their densities are greater than water, so they'll sink when released to the environment. Some of the health effects at low dose exposures, there's allergic sensitivity, there can be respiratory inflammation, oxidative stress, and suspected carcinogens. And so EPA does have some drinking water limits for some of these compounds, a microgram per liter for hexachlorobenzene, 600 microliters for 1,2-dichlorobenzene, and then a lot of these compounds are on EPA's priority contaminant list. And then on the right-hand side of this slide, I show some of the properties, thermodynamic properties of these compounds in terms of aqueous solubility, vapor pressure, and Acknowledgement water partition coefficient. And so as you increase the number of chlorines, so in our case, we're looking at mono, di, and trichlorobenzenes as the predominant ones that have been released to the environment. What you find is that the aqueous solubility goes down as you add chlorines, the vapor pressure goes down, but the hydrophobicity or the Acknowledgement water partition coefficient goes up. And so what will happen is that this will cause partitioning and sequestration in the subsurface. Just to talk a little bit more about the health implications, here's a little more information about that. So on the left-hand side, I've already mentioned some of these health effects. So at high concentrations, a lot of these compounds are known either to cause chronic or acute toxicity to both humans and organisms. And then on the right-hand side are some of the concentration levels which are important for these compounds. So they're on drinking water standards as well as on national priority lists and pollutant lists for treatment. And so at a lot of hazardous waste sites, we find these chemicals present. So what led us to this is there's a Superfund site not too far from us. It's called the Delaware, Standard Chlorine of Delaware site. This actually was made aware to us by USGS. They've been working on this site for quite some time. And this was a legacy manufacturer of chlorinated benzenes in 1966 to 2002. And over that operating period when they were in business, they had several spill events and particularly 2 discrete tank spills. So they were shipping this stuff out in rail cars. And they estimate over 2 million liters. And yes, that's right, 2 million liters of chlorinated benzene spilled on their property. And so this has gone through extensive remediation, largely to control the source zone. But it turns out there's an adjacent wetland. So this is a picture, the top picture is sort of the facility. Today, it's mostly barren land with a little bit of leftover buildings. But nearby is a Delaware estuary. And so we have wetland sediments, groundwater, and surface water issues associated with these compounds. And as I pointed out, primarily then it's monochloro, dichloro, and trichlorobenzene that have been spilled. So here's a little schematic of the hydrogeology of the site and the remediation challenge that happens. So on the right-hand side is this Delaware estuary, Delaware Creek, and the left-hand side shows you the facility. So it was very close to the water supply. And so what happened is when the chlorobenzenes were spilled, being DNAPLs, they sink in water, they caused the subsurface to be contaminated. And as groundwater flowed through it, it dissolved these chlorinated benzenes. And it turns out that the groundwater emerges at the surface water. And so we have a hyporeic zone where groundwater is emerging, and this is carrying contaminants into the surface water. And so we've identified an area of interest really at this groundwater-water interface is where we are looking at what kind of treatment we can do. It's a very large contaminated area. The groundwater has a lot of organic carbon in it, and so it's anaerobic in the subsurface. And then these compounds, chlorinated benzenes, because they sink, then you form these DNAPLs, and they form a source for a very long period of time. So what do we know about chlorinated benzenes and their biodegradability? Well, it does turn out that these compounds are biotransformable. But the way it works is that there's one group of organisms that is what we call anaerobic reductive dechlorination. is that they like to have compounds that have a lot of chlorines on them. So here on the left is the trichlorobenzene. And if there's an electron donor available, typically as hydrogen, then this can react with the chlorinated benzenes and pull off a halogen, or a chlorine in this case, giving you a dichlorobenzene. And then that can go again, sequence to monochlorobenzene, and eventually that goes to benzene. This usually requires a consortium of organisms. So the electron donor, ultimate electron donor, is usually organic carbon, here depicted in the lower left as a complicated substrate. It could be natural organic matter, it could be plant tissue, any kind of detritus. And then it goes through fermentation to produce short chain organic acids like lactic acid. And then this is ultimately fermented to produce the hydrogen, which goes into this sequence, so what we call reductive dechlorination. So anaerobically, the compounds that are polychlorinated tend to be more reactive as an electron acceptor. So they're accepting the electrons from this donor, and they're being reduced, which ultimately can cleave off the halogens. And then we know that chlorinated benzenes can also degrade aerobically, particularly the ones that have fewer chlorines on them. So here in the lower frame, you see chlorinated benzene. It can react with an organism that's aerobic with oxygen, and it cleaves it, and then you break apart the ring, and you get complete mineralization of the compound. So basically, we completely detoxify the chlorinated benzene this way. I'll point out that for the anaerobic reductive dechlorination, if you go from trichlorobenzene, di, mono, and benzene, that in many cases, these intermediates are still toxic, and particularly benzene, for example, is highly toxic. And so you really have not destroyed the toxicity or removed the toxicity, but you have transformed the chemical. So what we're doing is coupling these two processes. So if you couple anaerobic with aerobic degradation, then you have the opportunity to completely break down these chlorinated benzene compounds. And so the strategy is sort of depicted in this schematic here. And then the thickness of the little black arrow tells you the likelihood of that reaction occurring. So the thicker the arrow, the more likely it occurs. So as you go from a trichlorobenzene to a dichlorobenzene, monochlorobenzene with the anaerobic pathway, each sequential step is more difficult. It happens at a slower rate. And so the last step, chlorobenzene to benzene, is a very slow step. But if you couple that with aerobic degradation following the reductive dechlorination, then as you remove chlorines, you make it more reactive aerobically, to where benzene and chlorobenzene are highly reactive aerobically, and the trichlorobenzene and the dichlorobenzene are much slower. So by having a two-step sequence, we have the opportunity then to completely break down and destroy the chlorinated benzene compounds, no matter what, how many chlorines are present. And so our concept in the field is to create a reactive barrier. So here now we're showing that same location. We have surface water above the land surface. We have groundwater that is upwelling and entering that surface water, and because of these DNAPLs that are sediment bound. And then you produce, basically, rather than try to clean up the whole region, which is going to be very challenging, is to create a barrier, a reactive barrier that has these anaerobic and aerobic zones in them. And that's shown here. And so what will happen naturally is that the surface will be aerobic. The groundwater here is anaerobic. So as the water flows upward, it goes from the anaerobic to the aerobic zone. And this provides the opportunity for the trichlorobenzenes to be reductively dechlorinated, and then the monochlorobenzene to be oxidized to carbon dioxide and completely destroy the chemicals that are present. So how would we actually achieve this? Well, in the field, then what we would do is we would put in some sort of packing material, and the concept here would be also to have granular activated carbon mixed in as a sorbent to help smooth out fluctuations in the concentrations, and then some sort of solid carbon source. For example, crab shell chitin is known to be a pretty good carbon source for biodegradation reactions. And so in this way, if you would lay down a barrier that had this material in it, you would have internal food to drive their anaerobic reductive dechlorination, and then you would have aerobic oxygenase at the surface to drive the aerobic oxidation. And so with this kind of barrier, it would require low capital costs. You're not doing a lot of pump and treat, so there's a maintenance cost should be low as well. So there's a lot of attractive aspects of doing this reactive barrier. Now, we've also found that with these compounds at this site, that naturally there's not a lot of organisms present that can degrade these chemicals. And so what we've done, we've enriched cultures to do this. So there's an aerobic culture that we obtained from the groundwater that we've enriched in the laboratory. It happens to be from a well that was labeled 15B, so we call this culture 15B. It's maintained in the lab on these monodi and trichlorinated compounds. And so it's a mixed microbial community. And then there's an anaerobic culture we obtained from a laboratory called Cyrum Labs, and it's labeled WBC2. And this was developed originally for chloroethane and chloroethane degradation. And it contains some known organisms that degrade chlorinated solvents, dihalococoides, and dihalogemonones degraders. And so we're applying this now for the chlorobenzenes. So it turned out that this culture also can degrade the chlorinated benzenes. So in order to stimulate and develop this reactive barrier, we're bioaugmenting with these two cultures, one aerobic and one anaerobic. So here's the concept that we are testing in the laboratory. So right now we have a laboratory investigation underway. So again, on the left is the conceptual model. We're in the field, we have this anaerobic and aerobic zone. And to simulate that in the laboratory, we have columns that we feed anaerobic water with our chlorinated benzenes in them. And then we inject somewhere in the middle of the column, oxygen saturated water to create an aerobic zone. And so that's shown on the right-hand side, the actual column system. So we have a feed port basically where oxygen saturated water comes in to establish these two anaerobic aerobic zones. So here's further a little more detail about the column design. So these columns are 15 centimeters in length, and then about a third of the length from the top is where the oxygen water is injected. They're upflow columns, and so the feed at the bottom contains a trichlorobenzene, and then we're looking at transformations in the column, and then with the oxygen, we get the aerobic degradation. We have sampling ports located along the side so we can do vertical profiling of our contaminants and make mass balances with that. So Stephen Chow really developed this system, and a lot of credit goes to him to be able to operate these columns. Here's another schematic of the system. So the concentrations at the field site, I'll point out, are pretty high. They're in the milligram or tens of milligram per liter range. Normally when you find waste sites with chlorinated solvents, they're usually in the microgram per liter, orders of magnitude lower, but these are much higher because of the amount of chlorinated benzenes that were spilled. And so to simulate that, we've had to create a system that we can have tens of milligrams per liter going in. And so Stephen set these things up so that there are these Tedlar bags that in one bag he has the anaerobic dissolved chlorinated benzenes with the carbon source lactate, and another bag he has mineral media so he can prevent contamination and growth in these bags. And then this feed is mixed to feed the columns. And then there's a separate feed where he bubbles oxygen, pure oxygen, into water, and that becomes the oxygen feed to create the aerobic zone. So one of the main questions we've been looking at is what is the interplay between the electron donor, the carbon source you need to drive the anaerobic reactions, and the amount of oxygen you need, and the balance between the anaerobic and the aerobic zone. So it is a challenging system when you think about it to get all of these right. And so what has been done is in the laboratory system, these columns are packed with sand. There's a 6.5-hour residence time. The normal feed flow is 100 microliters per minute, very slow flow. And we're using lactate as a model compound because we know that lactate is a good electron donor for the reductive dechlorinations. And so the way this worked is we're feeding 1,2,4-trichlorobenzene at the bottom of the column with the anaerobic zone, and with various doses of the sodium lactate to simulate what a range might be in the field, but also look at what happens when you have both low concentrations and high concentrations to drive the reaction. So here the concentration of the trichlorobenzene is 5.75 milligram per liter. And then Stephen has operated these for 300 days at this point. So this is no rest for the weary, as we say, in this kind of operation. So it requires a lot of attention for the feed and attention to maintaining it that long. But here's an operational summary on the lower left that what we've done is varied the lactate and the trichlorobenzene ratio. So we keep trichlorobenzene fairly constant, and then we add different doses of the sodium lactate to simulate either a very low carbon zone or a very high carbon zone. Now I'll come back to that as we'll look at the column operation. So then what is done is at the bottom frame panel is that same picture of the lactate to trichlorobenzene. We can then look at what happened and do a mass balance. So we periodically collect samples, we measure the chlorinated benzenes, the tri, mono, and di, and monochlorobenzene. We also measure chloride as an indicator of demineralization. So if you take off a halogen, then you get mineralization that way. And so there's two panels that are shown. The top one in green represents the amount of mineralization that occurs. So basically, this is just a mass balance on how much chlorinated benzene we added in the feed in moles, mole per liter, and how many moles per liter goes out. And what that represents then is degradation overall of the compound. And then we looked also at the dechlorination process. So dechlorination is an important first step to achieving this oxidation under aerobic conditions. And so in this case, we're looking at the transformation of the 1,2,4-trichlorobenzene all the way to monochlorobenzene. And if that transformation is complete, then we'd say the percent dechlorination is near 100%. And then if it's incomplete, then it would be close to zero. And so the blue line, you see the trace where indeed some of the time it's zero and some of the time it's near 100%. Likewise, in aerobic degradation, sometimes it's very high, sometimes it's very low. And that's dictated by the bottom panel, which are our variables in terms of the amount of substrate that we're adding. So what we're going to do now is break this up into three kinds of segments. Well, this one segment is when we have the highest electron donor. So if we really pumped a lot of carbon in the system, what happens here? Well, it turns out when you have a lot of carbon, you get near complete anaerobic reductive dehalogenation. So that's shown by the blue line of near 100%. So basically the trichlorobenzene is going to monochlorobenzene, but there's so much organic carbon there that basically you use up the oxygen with the lactate degradation, and there's no oxygen left over for the chlorobenzene degradation. And we see that when you look at now a profile along the column. So in this figure, the distance is percent of flow path. So 0 represents the bottom of the column and 100 represents the top of the column. And so we have these 4 sampling ports for that. And we're looking at concentration. It's color-coded. So the trichlorobenzene is sort of the little light red, dichlorobenzene is green, and monochlorobenzene is blue. So what happens when you have a lot of lactate, you drive the reaction, so you get complete reductive dechlorination. So the trichlorobenzene disappears by the first sampling port, and then you create some dichlorobenzene, and then you get monochlorobenzene throughout the column. But you don't have any overall net mass loss of the chlorinated compounds. And that's shown by the release of the chloride, so that there is this, the black line and the dotted line represent chloride release, and the measurements and the theoretical agree very well. And so you get chloride release when you have reductive dechlorination, but then when you don't have any aerobic degradation, it just levels off. So too much organic carbon basically shuts down the aerobic process. Then we looked at the other extreme, which is a very low electron donor. So this is below stoichiometry, or maybe right at stoichiometry for the degradation of trichlorobenzene, but there's various competing reactions that are going on. And so when you drop the lactate down to very low concentrations, what happens is it takes a little while for the column to adjust. So it was working well for dechlorination, the blue line, but then after 20 or 30 days, then it drops, and essentially we have no dechlorination anymore. The aerobic zone is maintained, but it doesn't get enhanced. So you do see some aerobic degradation because now you have plenty of oxygen available. So oxygen availability is high when the lactate is low. And we see that here, that if you look again along the flow path, the trichlorobenzene no longer is degraded or reductively dechlorinated, and we don't see any appearance of mono and dichlorobenzene. But what is occurring, it looks like the trichlorobenzene is partially being degraded in the aerobic zone. So we are getting some release of chloride from the trichlorobenzene degrading. So we do know that it can degrade aerobically, but it is a slow process. And then finally, there's the intermediate carbon at 31 milligram per liter. So this case, then you get sort of stable mineralization. So the green line bounces around some, but you are seeing significant mineralization. And you also see anaerobic dechlorination. So now this is an intermediate range, and so we're able to get both dechlorination in the anaerobic zone and aerobic oxidation. And that's shown here. Along the flow path, we see that the trichlorobenzene declines because of the reductive dechlorination. We have appearance of dichlorobenzene and monochlorobenzene. And then in the aerobic zone, then we get these compounds degraded and release of additional chlorine. So this basically, in our mind, confirms the idea that you can sequence these biofilms, the anaerobic and the aerobic biofilms, and get significant mass removal through the combination of the anaerobic and aerobic processes. So Stephen, more recently, has then started to probe the microbial populations to try to look at the dynamics that are involved there. And so he's using molecular tools, molecular biological tools, to try to look at known degraders for some of these dechlorination reactions and look at some of the functional genes that are there. So he's been sampling. So what he did at the end of the 300 days, he sacrificed a column and then segmented it. And then each of the segments then he subjected to sequence analysis, a quantitative PCR, and then did the microbial community analysis with that by pyrosequencing. And so we have one set of data to show you at this point. He's going to probe this a little further, but on the upper graph, that's what I showed you before. So this is the sort of moderate lactate situations where we're getting good reductive dechlorination of the trichlorobenzene to dichlorobenzene and monochlorobenzene, and then aerobic degradation. And then at the bottom panel is superimposed on that flow path what he found for the microorganisms. And so the black line represents total bacteria. So we know that when you have a column system like that, this typically biofilms will be highest, or the amount of biomass is highest at the entrance point, where you have the food, most food coming in. And then it'll taper off with distance as you have less substrate available for the biofilm to grow. But more importantly, here are two organisms that are known to degrade or do anaerobic reductive dechlorination, this Dehalobacter and this Dehalococoides. And so in this case, we did not find any Dehalococoides. The green line is essentially at 0 across the flow path. But the blue line, this Dehalobacter, was high in the anaerobic zone. And then when you got to the aerobic zone, it tapered way off. And so this is at least indirect evidence or evidence that in the anaerobic zones of reductive dechlorination that we're now enriching for an organism that is known to cause that reductive dechlorination. So we're quite excited about doing further community analysis to confirm the reactions and know how stable the biofilm will be if it were implemented in the field. So some of the key points that we've done is that we've demonstrated that we can achieve through the column system degradation in a simple sand column system with these substrates available and bioaugmenting them. So we bioaugmented the anaerobic culture and the aerobic culture. These concentrations are very high, 5.6 milligram per liter for the 1,2,4 trichlorobenzene, and this is 17% of its aqueous solubility. So that's what we're dealing with at this site, is these very high levels. And we did find that the one advantage of a biofilm is that it's pretty resilient, in that if you do have changing conditions, it does adapt and you're able to maintain that biofilm. And of course, the key message is that there's this balance between anaerobic zones and the aerobic zone, the oxygen availability, and that's going to be tied to the electron donor that's present and the amount of oxygen available. And so you have to make sure that there's oxygen available for degradation pathways. And then if you have too much organic carbon or too much electron donor, in this case as low as 50 milligram per liter as organic carbon, this can inhibit aerobic degradation. So that's going to be the trade-off and interplay that you have to work on. I will say that in parallel, there have been some researchers at USGS that have done a field trial of this technology. And so they've actually gone out to the site and had little meter square zones where they put down this biobarrier with these organisms, and they operated for about 6 months. And they had positive experience. They saw some chloride release, and they were able to see enrichment of some of these organisms as well. So at least there's potential of actually exploiting this in the field. I will say there's a couple of more experiments left. Stephen is planning to graduate in about a year. And so we're wrapping up some of his research, and he's got a couple more chapters in his thesis to work on. And so one of those topics is to look at different electron acceptors. So he started some new columns, and he's going to feed them nitrate and sulfate to look at how the impact of the nitrate and sulfate will be on the anaerobic reaction. So normally the fermentation processes are what have been reported to be needed for this reductive dechlorination. And so if you add nitrate and sulfate, that might shut that process down. And we don't know that, but that's one reason why we want to try this, because at the field site, it's an estuary or has the ocean water influence. So there is some sulfate in the surface water. And so if you get the tidal action, you might get some sulfate into the sediment. And then there is some nitrate around too from agricultural activity in the region. And so we're just looking at those electron acceptors and their influence. And then finally, to better simulate what could happen in the field, is we're now moving to what we call sort of an open mesocosm beaker experiment, where we're trying to simulate better what's going on. So in the column that I showed you, we basically dissolve the trichlorobenzene in the feed. In this case, we're setting up these beakers where we're actually adding the chlorinated benzene as the neat solvent, so as a DNAPL, dense non-aqueous phase liquid at the bottom. We have a needle that allows the feed flow to come in and penetrate that neat material, the chlorinated benzene. And then it flows upward through sand that has chitin in it and granule activated carbon. So we can look at the carbon source coming from the chitin and the carbon, granule activated carbon absorbing some of the material. And then we'll bioaugment the systems, putting in both the anaerobic and aerobic cultures and relying on surface oxygenation for this. So we recognize that the flow path here is not going to be as well defined as in the column systems, but at least the physical characteristics, the location of the chlorinated benzenes, the presence of chitin and the presence of granule activated carbon are going to be more like it would be as a biobarrier in the field. And so we feel like at least some experiments probing different conditions here, and you see pictures of what they look like in the laboratory, will then give us a better feel for how the system will perform. Going from the laboratory to the field scale process. So that's basically our project. I wanted to acknowledge there's a lot of helpers we've had. So in my group, we've had a number of undergraduates and master's students helping out. We've also had other doctoral students that are helping give Stephen moral support and helping with the analytical process. That's Chris, Hannah, and Margaret. Mike Rose is in a different research group here, but he's helped with the analytical instrumentation to develop methods to analyze the chlorobenzenes. And then in terms of the pyrosequencing, the quantitative PCR, we have our environmental microbiologist, Sarah Preheim, and postdoc Eric Sikowski and Andrea Fraser, PhD student, helping out there. Juan is our lab manager here, and then 3 researchers are co-PIs based on a research grant we have from the National Institute of Environmental Health Sciences to do this work. And that's Michelle Laura at USGS, Amar Wadawan at Arcadis, and Neil Duran at Geosyntec Consultants. So we're grateful for their input. And then we have funding from NIEHS, as I mentioned, but also an NSF IGERT that funded Stephen for 2 years to help work with this project. So with that, I'll stop. And I know we have some time now for questions. I'm happy to answer questions that you might have.
Okay, thank you very much, Ed. That was an excellent lecture, really interesting. So questions from anybody, either using the group chat or your microphone, whichever you prefer.
I'll probably need to stop sharing to see the group chat, is that right?
Yes, please.
Okay. I see, okay.
Ed, here's Carson.
Yes?
I have a question.
Okay.
If I may ask.
Sure.
So you mentioned, you talked about mineralization, and as far as I understood, you mostly refer to compounds that don't have any chlorine. Is that correct?
Well, in terms of what you mean at the end, that come out.
Yeah. When you say something is mineralized, is that in your terms? Does that mean you have all the chlorine was removed from the compound?
No, it goes to carbon dioxide and water.
Oh, it goes. Okay, it does go to carbon dioxide.
Right. So the complete reductive dechlorination is removing all the halogens or all the chlorine. But
Right.
mineralization would be completely oxidizing to CO2 and water.
Okay, well, so my question was, what kind of intermediates did you see, or did you look for any intermediates between your benzene, for example, and carbon dioxide?
No, we did not. And so basically we're doing the mineralization by, at that stage, chlorobenzene, we're looking at the loss of chlorine,
Right.
chloride. So we really don't have, right at this point, we don't have a good way. We have not done a lot of mass balance on the intermediates between the two. We just know that aerobic degraders, based on the literature and previous work in my lab, they do oxidize these chemicals to CO2. So that's what we're assuming is taking place here.
Okay. All right. Thank you.
So I see there's a question about actually laying down the barrier and what happens with the roots and whatnot. And actually the plants themselves actually might be helpful because they can actually help put oxygen lower into the barrier approach. So when USGS conceived this, they were thinking about basically putting down maybe something that's 30 centimeters or maybe up to 50 centimeters thick. And if plants were growing on it, that would actually help at least the upper layer be aerobic. But of course, then the integrity of the barrier also has to be looked at. And so that would be something to have to explore. But when they did the trial run, they just cleared an area, a meter square, so there wasn't any vegetation on that. What about cost? Well, yeah, so that's, at this stage, USGS actually, this is a proposed remedy for the site. So this is a Superfund site, and they are actually, they've done the hotspot treatment, a lot of the removal of material. I think they've done some surfactant flushing, co-solvent flushing to remove some of the local chlorinated benzenes, but they are planning on doing it. So I'm not a cost person per se to know, but the costs are not, it's not that expensive. I know granule activated carbon is very frequently used in sediment remediation where they lay down these caps because it is one of the most low cost of any options that you have. And so chitin is pretty cheap because it's crab shells, it's readily available. So we think that the costs are not going to be that great. Certainly cheaper than digging it up and hauling it away or trying to use any kind of other kind of remedial technologies. Okay, so in terms of analytical support, I see there's a question. So chlorinated solvents, these compounds, chlorinated benzenes, can be readily detected in either an electron capture detector system on a GC, gas chromatography, so through an extraction technique, or you can also use a gas chromatograph mass spectrometer. So we use both. So we use GCMS frequently, but we've also used the electron capture. And if you want to do benzene, you can do a flame ionization. So they're volatile enough that you can use a gas chromatography as a basic strategy. So I think that so far the chat questions. Oh, yeah, how are the temperature? Yeah, so obviously temperature would be an important issue, particularly in the winter time. So if it's too cold, then these reactions are going to slow down quite a bit. Summertime, of course, they'll be warm enough to do. So we kept ours pretty much at room temperature in the laboratory. I would imagine that things would slow down quite a bit in the wintertime too. Although again, some of the dissolution reactions might also slow down some too. One of the benefits I'll point out about having the granular activated carbon there is that when there's biodegradation occurring, then you basically free up the absorptive capability of the granular activated carbon. So you can store chemicals in the granular activated carbon, and then later when the biodegradation kicks in, it can pull the compounds off the activated carbon. So with low temperature situations, the carbon could then be the predominant removal mechanism, and then when it warms up, the bacteria could kick in and basically replenish the activated carbon. So I will also say that even though we're using chlorinated benzenes as the model compound here, because that's what the site has in Delaware, we also think that the concept will work for other chlorinated solvents. We know that there are solvents like trichloroethylene and tetrachloroethylene that are widespread, and people are injecting dihalococoides, so there's a lot of bioaugmentation going on for those. So we may be able to use something like this for them. PCBs is another one, polychlorinated biphenyls, because those compounds degrade by reductive dechlorination. And then the chlorinated biphenyls, mono and di, could be oxidized aerobically. So we think that the strategy would work for a number of different chlorinated compounds.
Okay, well, if there are no further questions, I want to thank you again, Ed, for a really stimulating lecture. It's really interesting work that's going on. Oh, I see that there's a question has just come on the chat, so I'll let you do that one.
Right. So as I pointed out at the beginning, because this is a passive barrier, reactive barrier, we're not really cleaning up the source zone much. And so if the source zone persists, basically this could be something that has to be there for decades, if not longer, for that. So what it is, it's a passive remediation system to reduce the risk at low cost. As long as the contamination still exists on the site, then you would continually see these chemicals there. So the time would still be very long, but you are protecting the ecosystem and protecting humans that could be exposed to these chemicals.
Well, I don't want to stifle questions. These are good questions, so I'll give one last chance if there are any further questions. And while I'm doing that, I'll remind people that our speaker next week will be Chris Metcalf from Trent University, and it'll follow nicely from today's lecture because he's going to be looking at some ways to remove organic contaminants with UV. Okay,
so I see that. The second question, our follow-on was, could it be a more portable solution for remote sites? Yeah, so it's actually, it's not, as opposed to digging and transporting it. So yeah, so the idea is that by doing it in place, it doesn't take a lot of resources. It's pretty manageable. Activated carbon is fairly inexpensive, and this chitin would be, and so you can install it pretty easily. It doesn't require a lot of pumping wells or extraction wells, injection wells. So yes, I would consider it a portable solution. Of course, it's far north of Canada. It's pretty cold, so I know that there are people in Canada do what they call cold regions research on bioremediation. So you'd probably have to work on the kinetics and see if it would be sufficient to be able to achieve that. And again, the carbon could help maybe in the wintertime, and then in the nice summertime, you'd be able to achieve most of the biodegradation. But we do try to avoid the digging and transporting it out, that we call dig and haul. That's something that we don't like to do because they have to put it somewhere else, and typically it's a hazardous waste landfill, and that's very costly.
Okay, with that then, I'm going to say thanks again to Ed. Really appreciate your talk today, and look forward to having you all back next week.
Okay, thank you very much. I enjoyed the opportunity. Good.